Hello, and welcome to this tutorial on eDNA2obis, an eDNA data publication tool built specifically for fair eDNA data. My name is Baden Wilms. I'm a research engineer at NOAA AOML in Miami, Florida. And today I'm going to be showing you how to transform your eDNA dataset from a file that's on your computer or on your Google Drive and transforming that into a published data set on the global repositories of OBIS and GBIF. We're going to be running a real data set today, which was published using eDNA2obis, and it's a study targeting the potential prey of the endangered Rice's whales in the northern Gulf of America. And so today we're going to walk through what the tool does. We're going to take a look at the input files. We're going to watch it run. We're going to customize our run, and then we're going to review the outputs and then do a bit of a practice submission just so you can see what that looks like. So eDNA2obis can be found on the AOML Omics GitHub page, where you will also find all of our open source tools related to eDNA research. Now, eDNA2obis specifically was designed to reduce the number of technical challenges when it comes to eDNA data publication and make that process repeatable, reliable, and customizable. Its core functionality is to transform eDNA metabarcoding data into a Darwin Core archive, which is the proper input format for OBIS and GBIF. And I think what makes eDNA2obis unique is its configurable nature, as well as its transparency, showing users every step of the conversion, including the taxonomic assignment information. So these two reports, they're not submitted to OBIS and GBIF, but they're purely for users to understand what's being done to their data and to have process reports documenting that. These reports can help you find issues with your data. They'll give you helpful errors or warnings about your data, and they help make this process a little less painful. So the goal here is to take fair metabarcoding data and transform it into standardized biodiversity occurrence records and related metadata that can be stored on OBIS and GBIF. Don't worry, your input files are not edited by eDNA2obis. They're just simply read, and then your new files are created from scratch. And I will say, if the words fair eDNA do not ring a bell, it is the data standard that all of these tools are built on. And so please watch the previous tutorials that go over the data standard, how to make your data templates, how to fill them in, and then come back here when you're ready. So first things first, we're going to clone down the repository. And then once this is cloned down, we're going to set up our Python environment. There's two options for this. You can use whichever you prefer, but we have Anaconda or Conda, or you can simply do it with Python virtual environments. So opening this up, we can see here, this is the environment file for Conda users, and this would be your requirements.txt for Python virtual environment users. Today, we're just going to set up our environment with Conda, but again, you can choose whichever method you prefer. And I'm just going to rename this something different. I already have an eDNA2obis environment installed, so. And all of these commands that I'm running can be found on the eDNA2obis README. I really try to keep that up to date and clear for users. Great, and now that our environment's been set up, you'll want to activate it, and then you're ready to run eDNA2obis. But before we do that, we're going to take a quick look at our input folder, our Rice's whale prey dataset, which is an example dataset that's available to you when you clone down this repository. But taking a look at here, the main thing to point out is that there's really two types of data in this folder. There's eDNA metadata, so project, sample, experiment run, and analysis metadata, which are those fairy templates that you generated and filled in with data manually. So they're all here. And then you also have raw data. And so your raw data is going to be your ASV taxa features and then your OTU or abundance tables, both of which have feature IDs, and then the OTU table has SAMP names, and then your taxa features has your feature IDs as well as your DNA sequences and taxonomies. These raw data files for us in our workflow are produced by Tourmaline, which is our open source amplicon sequence processing workflow. But just note that you do not have to use that specific amplicon sequence processing workflow to produce these files. We just ask that your raw data be in this format, and then you'll be ready to go, ready to run eDNA2obis. We are ready to take a look at the config file. And now this is where the customization of eDNA2obis really comes in. This configuration file is really the engine of eDNA2obis. And once you're comfortable understanding this file, then you'll be ready to run this code yourself. So we're just going to take a look at this config file and just go through all the steps here. So starting off first, we have our config file path, and I recommend just leaving that in the root directory. A copy of your config file that you used will be put into your output folder so your results are reproducible. So metadata format, this was covered in the last tutorial, but you can select here either the NOAA Fairy data standard, which just has the analysis metadata in separate files, or you can do the generic or standard FAIR-E format, and that is also compatible with eDNA2obis. And then finally, we have our eDNA2obis run name, which will just help name things according to this specific run. So the output folder, the report, the copied config, stuff like that, just to help keep things organized. And I will say, one final note about that NOAA format is that is actually the input format required for the Ocean DNA Explorer. And this is our new eDNA data portal and visualization tool. And it's full of features that have been iterated on and requested by eDNA researchers. And it's just a really great tool to kind of explore your data a bit. It's got great UI for search. It's got a robust API that's well documented. You can make graphs on the actual website, as well as just browsing data. So we can see this Rice's whale prey data set on ODE itself. You can take a look at the taxonomies found, all sorts of great eDNA specific features. So I'm just going to plug that. All right, so on to step 2. We have some flexibility in the input formats. So combined metadata file, that just means if you have one Excel file that contains all of your metadata sheets in one, you can just do that and then specify the sheet names here and the file path. Or you can have separate metadata files. So combined metadata is false. And then as we can see here, we have separate TSV files for all of our metadata, and we put their file paths here. Additionally, you can customize the header rows of your input files in case you generated these input files with other tools. If you generated them with Fairy Sheets or Fairyator, you'll be in good shape. You won't have to adjust this. On to step 3. We've dealt with our metadata up here, so now we're going to link our raw data. And importantly here, you just go by each analysis, you specify your assay name and then your 2 raw data files. And then if you're in NOAA format, you're going to include your analysis metadata files here. So in this data set, we have 2 analyses using 2 different assays. And so for step 4, we have some control over our control samples. So whatever column you use to specify if that sample is a control or not, you give that column name here. And then you give the values which indicate if that sample is in fact a control or not. So those are our values. You can customize them. And that is because OBIS and GBIF do not want control samples in the submission. So eDNA2obis will remove these. On to step 5, a simple one. OBIS and GBIF would like a location ID field included in your data. The format for this is very open-ended. You can put pretty much whatever you want in there as long as it's related to location. So for us, we've got station ID and line ID. You can give this either one field or a list of fields, and it'll combine them automatically. And so step 6, now we get into our taxonomic assignment parameters. Now, this is probably the most complex part of eDNA2obis, and these parameters have been iterated on and adjusted as more people have used eDNA2obis, just trying to give useful features for people to use to help cut back on that amount of manual assignment and really just try to keep it automatic. The other thing to note is that OBIS and GBIF both require your metabarcoding taxonomies to be aligned to worms. That is the World Register of Marine Species. And you'll notice that for eDNA2obis, we support taxonomic assignment to both worms and GBIF. For now, you can only submit to OBIS and GBIF aligning to worms taxonomy. That might change in the future, but for now, if you're publishing, you'll want to align to worms. And so here, you can see you can pick either worms or GBIF, and then you'll have either GBIF specific parameters or worms specific parameters. And I won't go through them all right now. In fact, on the README of eDNA2obis, there is a lot of information regarding taxonomic assignment. It's a complex process, but there's a lot more documentation there. So I'm trying to find this. Yes, so here, this section here will give a lot more background onto what each of these parameters actually does in the code. So feel free to review that. And also, too, I would like to mention that if you only would like to run taxonomic assignment and not do the entire pipeline, that is also possible. There is a command line version of eDNA2obis that only does the taxonomic assignment called taxassign. And so the run commands are listed here. And that you just give it a file with a bunch of taxonomies, and this code will align them. So that's another useful feature. And so here, on to step 9, we get into our optional files. So these are not required for a submission to OBIS or GBIF, but we definitely recommend that you include them. The first of the 2 optional files will be the EMOF, or extended measurement or fact file. And this is used to attach standardized sampling protocols and environmental data to your DNA occurrence records. So how things were sampled, measurements like temperature, salinity, They can all be connected in a standardized way using this file. Now, this step requires manual effort, and there's no way around that. In order to format it, you'll want to follow OBIS's documentation on how to connect these measurements with your event data and your occurrence data. But essentially, what you're going to be doing is searching the NERC vocabulary server, finding the fields that you want to use, and finding the agreed upon vocabularies for those fields. And then you'll want to go in here, which is the input file for EMOF creation in eDNA2obis. And you're going to want to manually fill all of these columns for each of the measurements that you would like to connect. And this can be done either on an occurrence level or an event level. And then after the EMOF, we also have the EML, which stands for Ecological Metadata Language. And this file is essentially metadata for the project itself. And what we'll see when we actually go into the practice submission is that if you fill out the EML config file, which again is in the root of the repository, if you fill this in, when you go to submit your data to OBIS and GBIF, this file will actually populate a lot of the metadata fields that you would otherwise have to fill in manually. And so we have found this incredibly useful for large projects that contain multiple cruises. So a lot of that metadata is going to be the same across multiple submissions. Additionally, to fill in things like contact names that are often repeated, we have this contacts.yaml file, which you can use to have repeated contact information just to make that process easier. And so finally, we're on the last step of our config file here, and this is relating to short name. Essentially, enabling this parameter will split up all of the submission files by cruise names or short names. And so say you have a project that's one overarching umbrella project that has multiple cruises and you'd like to submit those cruises separately. Well, you'd enable this and all your input files, or all your output files rather, would be split by their expedition name. And so actually an example of this we can see on OBIS, some of this deep sea eDNA data from the NOAA ship Okeanos, which was published with eDNA2obis as well, was actually published as separate cruises under the same project. And so this short name splitting was very useful, saved a lot of time for projects that have multiple cruises. And so with that, we've gone over all of the steps of the config, and you should be ready to run your own data set with eDNA2obis. And so now, after all of that, we're finally ready to run eDNA2obis. We've set up our config file, so the actual run command is quite simple. And so this pipeline is going to do a lot of things while it's running. It's going to remove empty fields. It's going to align field names to Darwin core. It's going to create all your output files. You will also see the taxonomic assignment run with parallel processes. And the taxonomic assignment typically takes the longest amount of time, but this is a relatively small data set, so it runs pretty quickly. And then we'll also see here, we have our occurrence core created, we have our DNA-derived extension created, and then we have our EMOF, our EML, our HTML report, as well as our meta XML. And now that that has run, we can see that our output directory was updated, and there are our output files. You can open them up here. And so We have all the files we need for submission, as well as the config file that we used for reproducibility. And then we've also got our 2 report files. These are not to be submitted to OBIS and GBIF. These are purely for your reference as a user. So first, we'll take a look at the HTML report, which opens in your browser automatically after a successful run or after a run that errors out. And so we can see here at the top, there are some warnings. We have some fields missing with this data set. That's actually expected. There's a few fields missing, but yeah, overall, you can see every single step of the conversion pipeline as it happens, or I guess after it happens, but you can see every step. And so you'll get previews of, just the input data, how it loads in data. We can see it's removing control samples, and then every step of the way, it's going to confirm with you and just show you what it's doing. So you can clearly see that these are all control samples, and that looks correct. It'll tell you what columns have some NA values, because OBIS and GBIF, if there's a single NaN value in a column, it does not want that for submission. So it's just telling you what it's removing. And yeah, so this is the mappings to Darwin Core field names and how it's mapping each field. And then as it builds our output files, it'll actually give us previews of those output files as they get made, which is pretty cool. And then for taxonomic assignment as well, you get a lot of info returned back to you, including this taxa assignment info file. Now, this is another report file purely for your own reference, and we'll take a closer look at that in one moment. Yeah, after that, there's our DNA derived extension preview. As that gets made, it's letting us know some information about our EMOF file, a preview of that, and then our EML file. And so we could take a look as well at this other file that is given to us, which is the taxonomic assignment info file. And now this is super useful for when you want to review how eDNA2obis assigned taxonomies to your metabarcoding taxonomy data. And so all of the fields, there's a lot of different columns in this file. They're all described in the README of eDNA2obis. And this will tell you all sorts of different stats about why the code said that, this is the taxonomy that was assigned. It can tell you things like if there are multiple names for a certain species, selected match tells you if what was actually chosen, if it was an unaccepted name that got replaced, all kinds of things. It'll tell you ranks matched and ranks provided and how that correlates to an assignment score, the environment that species can be found in, even things like which API endpoint was used. It'll tell you that. So this can be really, really useful if you go into your occurrence core and see some taxonomies, some taxonomic assignments that you don't agree with. You can go into this file and see the alternate matches and why things were chosen. So this is proven to be very useful. That pretty much covers everything. You've got pretty much all the information you need to submit your own data sets to OBIS and GBIF. We can do one last thing and do a bit of a test upload to the Integrated Publishing Toolkit, or the IPT. Now, not just anyone can do this. You'll need to coordinate with OBIS and GBIF in order to get permission to published data sets. But should you get that permission, it's going to look something like this, and um, we can fill that in. It's an occurrence data set. We can create. And then you'll see here in source data, it allows you to choose files. And so then you would just go along and upload all of the files that are included in the occurrence core. And we can upload them. The EMOF file is rather large. And bam, we can see we have just taken a FAIR-E metabarcoding dataset from a file on your computer or on your Google Drive and transformed it into an OBIS and GBIF dataset. So I'd like to, if you're still watching this, I'd like to thank you for watching the tutorial. And just to wrap up here, I would like to say that this process today was, this data set was already uploaded. We didn't run into any issues. I will just say that for things like taxonomic assignment, you're really going to want to review those manually, and you're not going to want your published data to go online without reviewing everything manually. So that's why we've given the HTML report and the taxonomic assignment info report, so that you can check everything and make sure it looks okay. So with that, I'd like to end the tutorial and thank you again.